Hello. So this is the second part of second part of the 10-part series. So last time we did DNA, right? And protein synthesis. And so the function of DNA and protein synthesis and why we need DNA. We said DNA, like we said, it codes, it codes, right? It helps with the coding, so with the genes, right? So now we're going to continue. So, like I said, remember when we did DNA replication, right? I said DNA replication, you do it for two reasons, right? And like you do it for one main reason for cell division, right? So when the cell is about to divide, then DNA, um, there is DNA replication, right? So there's two types of cell divisions, and of course, these cell divisions they depend on. So these cell divisions they depend on why the cell is dividing, right? And where exactly does it take place? So these two types of cell divisions are called mitosis, and the other one is called meiosis, right? So mitosis takes place basically anywhere really in the body, right? Um, except near, so except in inside the sex organs, right? So however, mitosis happens like everywhere because it's just a cell division, it's just for growth, right? So it happens basically everywhere. And but the type of cells that it happens to are called um so the type of cells that it happens to I think they are called somatic cells, yeah. Somatic cells, right? Do you see? Cells in the living organisms, excluding the male and female gametes, right? And then sex cells, sex cells are the ones that that divide via meiosis, right? So those are the ones that divide via meiosis, right? And it's very important to know these like the difference between the two, right? Because in the somatic cell, so during mitosis, the reason why mitosis happens is mainly for growth, right? So to replicate that cell for growth, so that there's more of that cell, right? Right. However, so for the gametes, for the sex cells, right? Those ones we don't do them for growth, right? It's done so that basically you have reproductive material, so that during reproduction, like basically those two cells can fuse, those two sex cells can fuse, one from the male, one from the female, right? So meiosis happens for the explicit reason, for the main reason of reproduction, fertilization, right? Otherwise, fertilization cannot occur without meiosis, right? And then mitosis is just replication, right? So let us think about it properly. Remember when I did DNA, I said half of your genetic material comes from your mother and half from your father, right? And that happened during what? During so right now we are not gonna do mitosis, right? Because mitosis I think you're already done in grade 10, right? However, you'll see the process between mitosis and meiosis is very similar, and I will go through the differences towards the end, right? So we can continue. So now we are doing really meiosis, right? So um with meiosis, like I said, your genetic material half comes from your mother, half comes from your father, right? And that happens obviously during intercourse. And then basically what happens is that the reason why it's half half, right, is because of this meiosis, right? Do not forget when so during mitosis, right, when the cells replicate for growth, right, both the genetic material of the father and the mother basically it becomes duplicated, right? Because you have your genetic material from your mother and your father, both of them gets duplicated, and then you have basically an identical cell that you had, right? But with meiosis, that doesn't happen, right? Because don't forget, one cell has genetic information basically um doubled, right? It has double genetic, like double the genetic information, right? So therefore, during meiosis, what happens is that we have to halve the genetic information, right? We have to divide the genetic information so that what happens during reproduction, half of the genetic information that you have can meet with half of the genetic material from the father, right? So that's the reason for meiosis. That's why we do it, right? Then you'll see as I move on, like I said, introduction is very important to know why we are doing this. As we move on, do you see, like I said, everything is chained up, like in life science, like it connects. You see, started with DNA and DNA, but we ended at DNA replication. We didn't like move forward as to what is DNA replication. Now we are going from forward from that, right? So DNA replication, where does it happen? DNA replication, it happens in a phase called interphase. Now, interphase, you find it both in meiosis and mitosis, right? So when the cell is not dividing, right, what happens to this DNA? This genetic material, right? You'll see DNA is sometimes called genetic material because it does contain genes, like, right? So this genetic material forms a what? A tangled chromatin network, right? So when I'm talking about chromatin network, it's just that genetic information, that DNA basically wrapped up into a network, right? So that network is called a chromatin network, right? So that chromatin network during interphase, what happens? The DNA there, it replicates, right? DNA replication takes place, right? Right? Because we have this single strand during chromatin network. To, if there is no cell division, we just have a single strand of this chromatin network, right? So it's a network, it's a strand basically, right? You know the strand. So a strand is something like this, and then we have this network, right? This network, it's something like this. However, it's single stranded, it's a single stranded network. Then what happens is during DNA replication, you remember what happens, right? We get basically two strands, right? And then this single strand chromosomes become double stranded. So basically, they become double stranded, that tangle chromatin network, right? Then okay, so that's basically what happens during DNA replication. Now we can start with the process of meiosis, right? So like I said, meiosis, why do we do that? We do it for this gametes, right? For gametes is basically sex cells, right? And sex cells, what do they have? Half of the genetic material, right? So do you see, if meiosis doesn't okay, gametes will not be formed and sexual reproduction will not be able to take place. Why? Because your cell will have the genetic material of both. Basically, it will have genetic material of basically of you from your mother and your father, right? So which gives you really a full cell. But do you see that a full cell cannot be joined, right, with another full cell, because basically we need half genetic material so that those half, they combine to form one. Do you understand? So without meiosis, that won't happen, right? So you'll see what I mean. So now they say during meiosis, haploid gametes are formed, right? So haploid just means it's one, it's not two, right? So basically these gametes are, so basically this is important because the number of chromosome is not doubled when fertilization occurred, right? So instead, two haploid gametes form to fuse a, a diploid number, right? Right. So don't forget, haploid, it means half, right? So haploid is a genetic material like halved, right? And then diploid is the full one, right? Because diploid basically, it has genetic material from two. I get di means two, right? Double, right? So it has double the genetic material, meaning one from the mother, one from the father. Whereas haploids is just half of the genetic material, right? So um if we're talking about chromosomes, it will be 23 for a human, right? And then diploid will be 46, right? Then meiosis ensures the same number of chromosomes within a species remains constant from generation to generation. Do you see? So what meiosis ensures is that basically the organism itself remain the same, right? Because obviously, if suddenly we have more and more chromosomes, we are going to move away from being humans, right? Like our bodies. So the chromosomes have to be the same unless it's an anomaly, right? So, or a mutation. So what happens is these chromosomes within a species, they have to be the same. Of course, the genes within it, they can change, right? And as a result, you might change, like you might have physical attributes that maybe your father or your mother didn't have, or basically you can have them better. right? So they can improve. However, you will still be a human still, so you'll not have extra feet or whatever. So the genetic material, like, still that composes a human is still the same, right? And then in animals, you see meiosis occurs in the sex organs, right, to produce gametes, right? Gametes basically is sex cells, right? And then in plants, in spores. So spores are basically the sex organs of plants, and then which are used for reproduction in mosses and ferns, right? Then in flowering plants, meiosis takes place in the anther and ovule. So basically, what you have to, even in plants, right? So in plants, so basically these spores and the anther and ovule is basically the reproductive organs of the plant, right? So meiosis happens in the site of reproduction, right? And then, okay, process of meiosis. So know that a human has 46 chromosomes, right? Arranged in 23 pairs of homologous chromosomes, right? So now, okay, so for meiosis, there are two divisions, right? So the cell undergoes two meiotic divisions, whereas for mitosis, there's only one division, right? So for meiosis, it's two cell divisions, but for mitosis, it's going to be one, right? So, and they have like functions, right? So meiosis one, that first cell division is called, it's a reduction division basically. So meiosis one is a reduction division which reduces the diploid number, right, of chromosomes to haploid. So like I said, diploid is a full, right? So diploid is a full, right? um full organism or full human being, right? So it has genetic material from both the mother and the father, making it 46 for a human, right? However, haploid means half, right? Half. And this half basically comes from one, because you only have genetic material equal to one, basically, one contributor, right? So which is basically 23 in that case. And let's start with meiotic division. So although meiosis is a continuous process, events are placed into phases for convenience. Yes, so this process happens continuously, right? However, we break them down into these phases so that we see exactly what's going on, right? And then, like I said, when you're doing these types of questions, especially for life science, and this also, of course, works for um, maths, of course, paper too, because you need an imagination. But for life science, it's very important to have a very vivid imagination for life science. So I'd suggest that when you're doing life science, like, draw a lot of diagrams for yourself, right? Explaining the change in that, like how that thing happens. Like I said, life science is physical things, right? Unlike physics, you see physics, the reason why, of course, again, it's hard is because you're working with like forces, something that you can't really see. You have to measure it. You can see its effect, but you can't see it really. Life science, it's simple because you can see these things. You can see there's the cell, it changes like this, right? This is the change that occurs. So like I said, draw diagrams and see how the diagram changes, then you'll see exactly how it happens in your body, right? So what I'm saying is, do you see, every time when I explain these processes, I always want to explain it in a story format, right? So that I explain it sort of like in a process, right? Where everything, like, so that I see where everything fits. Same thing, please, like, do that. So it's either you take two approaches with life science, ne? It's either you do the story approach, so you tell yourself a story like I'm doing right now, um, from the start to the finish, know that story, or via diagrams, right? Via diagrams, because via those diagrams, you can tell those stories via those diagrams, or you can use them both. In fact, if you use them both, it will work to your advantage better, right? So let's start with the first meiotic division. So the first meiotic division is prophase one, right? So you'll see it's prophase one because it's meiotic division one, right? So what happens is, let's now see how that cell looks before we even reach to prophase one. So during interphase, right? So what happens is, they're telling us here that during prophase, the membrane and sorry, and the nucleus start to disappear, right? So what does it mean? It means that during interphase, we had a full membrane, right? And a full nucleus, right? That's what it means. And then they say the centromere starts to split, centrosome starts to split, right? So we have, which means we had a centrosome here, right? We had this. I'll just So it's a centrosome, right? Centrosome. And please, like when you're drawing these diagrams, label them, right? So there's a centrosome, and then okay, we have, which means we still had a chromatin network. Do you see that thing about a chromatin network? So this is a chromatin network inside the nucleus. And then, okay, so that's basically what we have, right? Before this phase. Do you see what I mean? I want to see where it starts so that like I know exactly how the process or what happens to the process. So it's simple here, right? During this first prophase, what happens firstly? We know that this thing, the nucleus and the nuclear membrane, they start to disappear. So how do you illustrate that this thing starts to disappear? With these dotted lines, right? Do you see that it shows that this thing is starting to disappear now, right? And then, so the nuclear membrane is this part, and the nucleolus, basically, it's this part. So this whole part is starting to disappear, right? This border basically starting to disappear, right? And then they say the central soul, so right, the centromere, I mean the centromere, this it splits right into what? Two central souls. So imagine that this thing is splitting, let's say it splits into half, right? And as it splits, do you see that it was attached via what? These spindle fibers. So as it opens up, so as one part of it goes up and one part of it goes down, what happens is we start seeing these spindle fibers, right? That this centromere was actually attached by these things called spindle fibers, right? So, and these spindle fibers remain, right? Right. So the centromere splits, and it splits into what? What are these two things called? Central soul. So it splits into two central souls. What is a central soul? It's basically half of a centromere, right? That has split and is attached via these spindle fibers, right? Do you see? And then, so now we know exactly what happens to even this one. So we know what happens to this one and this one. So what's left? The only thing that's left, you see, it's the chromatin network itself, right? So what do we know? This chromatin network is double right now because of DNA replication during interphase, right? It's doubled. And what happened is that this chromatin network, what does it do? It condenses, right? Do you see this is still a network, but it's a double-stranded network. So this double-stranded network, it condenses into what? Into these chromosomes, right? And it condenses into chromosomes. So chromosomes, what is a chromosome? The chromosome is, it contains obviously genetic material. So basically it's DNA, there's chromosomes, but these chromosomes, like I said, they are double-stranded because of DNA replication. So they have these two um they have these two so these two chromatids, and this thing basically is also, I think, called a central. Let's look. So these chromatin networks, they condense into so they condense into chromosomes, right? And these are chromatids. I just wanna make sure if this thing is a centrosome. I just wanna see what this thing is really. Okay, yeah. So this is also called so this is a centromere, right? So do you see the words here are very similar. So a centrosome is this, right? This is a centrosome which splits in which splits basically. So this is like I said, this is a centromere which splits into two centrosomes, right? And then this is a centromere, right? So this is a centrosome splits into two centrosomes, right? And then this is a centromere, which is basically a region where two chromatids, right, are joined. And this region, so this whole thing is called a chromosome, right? So what happens is that chromatin network, it condenses into this, but how many of this? Because we are human, it condenses into 46 of this. Obviously, if it's another organism, it's going to condense into um something other than 46, because other organisms have another number like of chromosomes, right? So after it forms these, it condenses into these chromosomes, right? You remember that these chromosomes, like I said, it's these chromosomes are made of genetic material, right? Right, from your mother and from your father, right? So genetic material, right? They actually affect certain parts, right? Certain parts of your body, right? These chromosomes, right? And then what they do is that 23 will affect certain part of your body, but another 23 will affect the same part of your body. It's a matter of which gene is dominant between the two, and you do that basically during genetics, right? So what happens is, now that you have 46 of this chromosome, do you agree that 23 of it will be from the father, 23 will be from the mother, right? Representing genetic information basically from the mother. And what happens is that when these things now they form these chromosomes, right? It's 23 from the mother, 23 from the father, right? So what happens is that, do you see that the reason for it is because each chromosome, right? So each chromosome, like I said, it affects a certain part of the body, right? So obviously, if the father had that chromosome, the mother will also have that chromosome, right? So what happens is that these chromosomes, now that they are 46, right? The homologous chromosomes. What is homologous? Like I said, the chromosomes that affect obviously the same gene, right? Those are homologous chromosomes, and they will lie next to each other, forming a bivalent. So bivalent is something like this. So let's go to the real definition of bivalent. So pair of chromosomes which lie next to each other and are physically in contact with each other at a point where crossing. So this is what bivalent means, right? So next to each other, physically in contact with each other, right? So that's what will happen with these two chromosomes, right? Do you understand what I mean, homologous? So they have basic. So when they say homologous, basically it's like I said, they control the same gene, right? So homologous chromosomes, so a pair of chromosomes, pair of the same shape, size, having similar genes for the same characteristics, occupying the same position. Do you see? So similar genes, do you see they affect similar basically positions in your body, right? So maybe eye color or something like that, they will affect something, right? And then what will happen is that those homologous, right? Because they affect the same, they lie next to each other, forming a bivalent. And bivalent basically just means that they physically lie next to each other, touching each other, right? And where do they exactly touch each other? How do they touch each other, right? So the inner chromatids, right? The inner chromatids from each chromosome, right? So so this is how they look, right? This is how they look. So this is one chromosome, right? So this is how they touch during this thing, right? So when they say bivalent, this is what it means. This is how they touch, right? So they overlap. So the chromatids, one chromatid overlaps over another chromatid, right? And then the point where they touch, it's called a chiasma, right? Or plural is chesmata, right? In the process called um, crossing over. So do you see that this chromatid crossed over to this chromatid, right? Right? So, and this point here, this process basically is called crossing over, right? So what happens during this crossing over? The genetic material from this one will be passed to this chromosome, and the genetic information from this chromosome will be passed to this chromosome. That's what happens. Do you see that? That's what happened here. This part went here, and this part went here. That's what happened during crossing over, right? And then do you see that this process brings about variation? Number one. So this is the part number one that brings about variation. Why does it bring variation? Because these chromosomes, although they come from your mother and your father, now they are fundamentally different. Because maybe this chromosome that comes from your mother, right? This maybe this purple one comes from your mother, right? It has this genetic information, right? Even though it's a small part, from what? From the father, right? And then as a result, this brings about variation. This and this is not the same anymore, right? So that's what happens in prophase. So we know exactly now what happens in prophase, right? So that's what happens in prophase. So we can move on, right? So the next one is metaphase. So metaphase, what happens is, now what happened in the prophase? We know that this basically was starting to disappear. So what happened in metaphase? This thing, it disappeared completely. Do you see? Here you show that it's starting to disappear. Here it disappeared completely. So now it's gone. The nuclear membrane, nucleolus is gone. So we don't need to talk about them anymore, right? Here. So here you just say, yeah, you can say that they are finally gone, right? Can even put that. And then what happens? Né? The first thing that happens you see is that the chromosomes, these chromosomes that are there, they're the bivalent ones after this has happened, right? They're gonna move to the equator of the cell. So they're gonna move to the equator of the cell, right? Right? And then the two homologous chromosomes, right? Don't forget these homologous chromosomes, right, are in a bivalent, right? So they're still in this bivalent. This bivalent basically is going to move to the middle of the chromosome. So as a bivalent, it's going to move together to the chromosome. Do you see that this one and this one are still the bivalent? They're still the same bivalent, right? And then what happened is that the two chromosomes, right? Um, the two homologous chromosomes lie in opposite sides of the equator, parallel to each other, right? So do you see that these ones? So basically, we have a cell, right? And what do they mean when they say equator? Equator means center of the cell, right? And when they say opposite sides of the equator of the cell, that means that let's say we have something like this. Now, let me show you right here. So this is basically the equator of the cell, right? This line that I'm drawing here, this halfway point. So do you see that even though basically these two are bivalent, right? Are bivalent, right? They are bivalent, but one of this homologous chromosome is in this side of the equator and one is in this side of the equator. That's what they mean here. Do you see? The homologous chromosomes move to the equator, right? They move here to the equator. The two homologous chromosomes lie in opposite sides of the equator, right? Parallel to each other. So do you see they're parallel to each other and they lie what? On opposite sides of the equator. This one is in the side of the equator. This one is at the side of the equator, right? And then which chromosomes lies on which side of which chromosome is totally up to chance, right? So what does it mean? It means that let's say we have this bivalent, right? And then we had this variation, right? Let's say this one, like we say, this the father, this one is the mother's, right? The purple one. So what happens is that, do you see that this and this may be like this, but the next pair of homologous chromosomes, maybe the fathers might be this side and the mothers might be this side. So that also causes another random arrangement. Do you see a further arrangement? And this further arrangement brings about variation, right? Because fundamentally, these cells will not be the same, right? So like I said here, so what two processes bring about variation? Firstly is the crossing over, right? Because it's not the same after, and also this variation, because that cell and that cell will not be the same, right? And then each chromosome in the pair becomes attached by by a spindle thread by the centromere, right? So what happens is, now that we know that these have how they've moved, right? The only thing that's left is the centrosoles, right? So what happens to the centrosole? The centrosoles, you explain that they move, right, to opposite sides of what the equator, right? They move to opposite sides of the equator, right? And the spindle threads, right? Or the spindle fibers, right? They will attach. So those spindle fibers will attach to the centromere, right, of every chromosome, right? So they will attach to the centromere of every chromosome. This will attach to this, right? This attaches to this centromere, right? And this centrosome will attach to this one, right? And this one will attach to this one. So this the centrosoles, right? So that's what happens. And you'll see this happens for a reason. Do you see that this one has to attach to this one? This one cannot attach, so this central soul from this equator cannot come now and attach, this spindle fiber cannot come and attach to this one. No, right? Because it doesn't make sense. In the end, this thing has to attach to this because it has to be pulled up by this one. Do you see? So this one cannot pull up this one that's from this equator, right? So that's what happens in metaphase. Metaphase, so they just go to the middle and then the spinal fibers are attached, right? And then we go to the next one. The next one is anaphase, right? So what happens in anaphase? Like I said here, in metaphase, these things are there, right? And these spindle fibers, the reason why they're attached to these centromeres, right, is to pull them, to actually pull these chromosomes. Do you see that during anaphase what will happen? One whole chromosome from each pair is pulled to opposite side by the contracting spindle fibers. Do you see? These spindle fibers are there for a reason. Now that they are tested, they pull. It's like they're fishing, right? They're fishing for these chromosomes. Now they are pulling it back in, right? This separates the homologous chromosomes to one to each pole, right? One to each pole. That's what happens. So do you see anaphase? Not really complicated. That's what happens. And then telophase, right? We have telophase one. So telophase, what happens is that now you uh it's basically we are forming the new cell, right? So a new nuclear membrane forms around the group of chromosomes at each pole, right? So do you see now, because this is poled, right? What will happen is that a new nuclear membrane will start forming here. Do you see? They're showing that the new nuclear, now that this thing is pulled, a new nuclear membrane starts to form now in these, so surrounding these chromosomes, right? And do you see that this central soul will actually be the central near. Now this central soul, right, will actually be a centrosome, right? Will actually be a centrosome. Do you see? It will be actually this in the new cell, right? So this and this pulls there, and then the nuclear membrane starts to form, right? The nucleolus returns to, right? Because the nuclear membrane starts to form. And then what will happen is that this is very important. Cytokinesis happens, right? So the division straight of the cytoplasm. Do you see the cytoplasm starts to divide? So the cell itself starts to divide, right? Right. So division of the cell splits the mother cell into two daughter cells. Do you see? Now this thing is split into two cells, split into two cells. Now we have two cells, right? And now it's important that to know that, do you see that in these two cells we have what? Half of the genetic material. That's why they call the first meiotic division what? They call it the reduction division, because it reduces the number of chromosomes in each daughter cell. So it went from 46 to 23, right? Then we move on. So the second meiotic division takes place in both daughter cells forming. So it takes place in both daughter cells forming. So it takes place in both daughter cells formed during meiosis one, right? So this one is not a reduction division. Um, it's not a reduction division. It just creates more daughter cells, right? So what happens is during prophase two, something similar will happen, right? Like I said, this the central some will start to split into two um central 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 centrals, right? It will split into two centrals. These two centrals, like I said, they are connected by these spindle fibers, similar to prophase one, right? Right. And then, so this is basically the same thing, and then we'll move on to metaphase. All happen in metaphase. So prephase one, the only thing that's different here is that you remember in prophase. So in prophase two, the only thing that's different from prephase one is that, remember, in prophase one we had a chromatin network, right? Here we have chromosomes, right? And then so these chromosomes don't, do you remember that? So we don't need to form so we don't need to condense the chromosomes. They're already there. They are 23. However, what you need to not forget is that they are 23. So they will not form bivalent pairs, because there's no pairs anymore. They're not paired, right? Half of the genetic material has been transferred to another cell, right? You see. So what will happen is that there are no homologous chromosomes here, right? There are no homologous chromosomes, right? So what will happen is that these single chromosomes will start to align. So will align in the equator, right? So they will arrange themselves in the equator, obviously randomly, right? Arrange themselves in the equator randomly, right? So whether this one faces that side or this one faces that side, it doesn't really matter. And do you see that as a result, this will also create variation, right? Because this part of the chromatid is not the same as this part of the chromatid, because this one, it already had crossing over during prophase one. Do you see, like I said, imagination here is very important. Do you see that this one definitely, let's say I get this cell will also form two cells, right? The two cells that will form will not be the same because this chromatid will go to that side and this one will go to that side. But you see that this chromatid is not the same as this one, right? So arrange themselves along. So that's what they mean when they say randomly. So this side can be that side or that side, right? And then do you see that the similar things happen to metaphase two? But what happens is that the firstly bivalent, so the bivalent pair, right? The homologous chromosome pair, it didn't come to the equator, right, as a pair. So they were just single chromosomes aligned, 23, right? Aligned. And now what happened is that sorry, these spindle fibers, right? They are gonna attach themselves, right? They're gonna attach themselves to each chromatid this time, not the centromere. Each chromatid, right? Because it's pulling out the chromatid. If it's connected to the centromere, right? Then you see it may pull the whole chromosome. So it connects here to each chromatid, right? So what happens during anaphase 2, the centrole is able to pull the chromatid via the contraction of the spindle fibers, right? So they will go to each pole, similar phase. And what happened during telophase? Telophase is that so the same thing will happen is that the the nuclear membrane will start to form and nuclear holus will start, right, to form, and then the cytoplasm will start to split. That's called cytokinesis, right? And then as a result, you have four, I mean, four cells, right? You have four cells that have 23 chromosomes, right? Because of these chromatids. So we have four cells, right, that has half the genetic material, right? Now, this second meiotic division is similar to the actual mitosis, right? Because what happens during mitosis? What happens during mitosis? During okay, we'll come here. So do you see here you have four different cells, right? These cells are not identical, but during mitosis, those two cells they become so the two total cells during mitosis they become they're identical, right? These ones are not identical, right? Then you'll see that this happens now, this process happens both in the females and males, right, of animals. Then during reproduction, one half will meet with the other and form a zygote, right, with 46 chromosomes, and then from there on mitosis will take place. That zygote would start multiplying and then forming a baby. Now we'll talk about that one when we go to human reproduction, right? So okay, so I just wanted to show you this diagram. So do you see this is what happens during mitosis, right? So during prophase, they don't form pairs, right? During this prophase, all of them will just line up individually. All 46 will line up individually. And then so the mitosis is similar to meiosis 2. So mitosis is similar to meiosis 2. But with mitosis, it will give you two identical cells, right? Why does it give you two identical cells? Because during this prophase, there is no bivalent pair, and as a result, there is no crossing over. So number one, there is no variation there, right? And then these chromosomes, they align, all of them, um, at the equator, right? Don't remember, don't forget, I mean, these chromosomes, right? Because there was no crossing over, the two chromatids, right, are exactly the same, right? So what happened is that after these things, they line up individually, and this thing actually contracts and pulls one chromatid, right? The chromatid impulse from this side and that side will be the same, right? And as a result, it will give you two identical cells. Now, with this one, this is not possible. Why? Number one, because of the crossing over, and number two, because of the random arrangement, right? But the main thing is the crossing over, right? And that's basically meiosis. So there's nothing really hard about meiosis, and you'll see even these differences are the same thing that, like I told you. You see, so I already talked about all of these things. And I think, but I did send you this textbook, ne? So please tell me if I didn't send you the textbook, because I think this is a really good textbook to use for life science, right? And yeah, um, send me any questions if you have any questions.